Today's video I will discuss about the blood groups or the blood grouping. In our previous video we all discussed the blood and the types of blood cells and the structures and also the functions of blood. Okay, then today discuss the blood groups. Okay, blood of a person is not always compatible with that of another. Hence, before the transfusion of blood, it must be tested. Okay, for its compatibility with the acceptor's blood. You all know this process. We all transfusion of blood, it must be tested for its compatibility with the acceptor's blood. Okay, only compatible blood is to transfused. Only compatible blood or same type of blood grouping will be transfused. In the compatible blood is transfused, agglutinations of the erythrocytes occurs, which leads to hemolysis. Okay, the membrane of erythrocytes contain antigens called agglutinogens. You all know the membrane of erythrocytes contain antigen and it's called agglutinogens. And the people have natural antibodies, that means agglutinins. Whenever there is an injury to a blood vessels, there is formations of rough surface. The inner smooth lining of blood vessels is disrupted. After injury, you all know the blood vessels of the inner lining of the blood vessels are disrupted. When the blood platelets come in the contact with such a rough surface, they are injured. Due to injury, they release the substances called thromboplastins. Okay, then the injured tissues also secrete the thromboplastin. The injured tissues also secrete the thromboplastin. In the presence of thromboplastin and calcium, in blood plasma, prothrombin, naturally occurring the plasma protein, is converted into thrombin. Okay, prothrombin plus calcium and prothrombin and calcium and thromboplastins is form the thrombins. Okay, which helps in the conservation of fibrinogen to fibrin. Okay, then the fibrin is insoluble and forms threads. Fibrin is insoluble and then it forms threads. The threads of fibrins are form a net. In the holes of this net, blood cells are entangled. Agglutinogens are genetically determined and are of two types. Okay, one is A or another one is B. It is possible to have antigen A in the red cells group A or antigen B in the red cells group B, or both A and B antigens at the red cells of group AB, or no antigen in red cells is group O. Okay, agglutinins in the plasma are globulins are of the positive type. Thus, the type A blood contain B agglutinin and the type B blood contain A agglutinin, while the type AB contains no agglutinin and the type O contains both A and B agglutinins. Okay, the compatibility of blood groups is shown below. Okay, now see. While determining compatibility, RBCs of donor must remain intact. That means their agglutination due to acceptors agglutinin must be avoided. Agglutinations of acceptors RBC due to donors agglutinin is not to be considered as it doesn't happen. Okay. The blood group A, B, AB and O. You all know these four types of blood group. The agglutinogens in RBC is A and the agglutinin in plasma is B. Blood A group. Then the blood group B, the agglutinogens in RBC is B and the agglutinin in plasma is small a. And the blood group AB, the agglutinogen in RBC is A or B, or agglutinin in plasma is no agglutinin. Blood group O, the agglutinogen in RBC is no agglutinogens and the agglutinin in plasma is A and B. Okay. Then saw these structures, blood groups showing their compatibility. Okay. See this. See these structures, and then the blood group A is donate the blood group A and also receive the blood group A. Okay. In these structures you just see the in our O is the universal donor. O is donate all kind of blood groups, blood group A, blood group B and blood group AB. Okay. And our AB is the universal receptors. The AB is receives blood from A blood group, B blood group and also O blood group. Okay. And the B is receive blood from B and donate blood from B and also receives the blood from O. Okay. You will see these structures, and now discuss about the thus the group AB is the universal acceptors and the group O is the universal donor. You all see the structures and then study. There is another antigen called Rh antigen present in some person's blood. Persons with Rh antigen in red cells are called Rh positive and those without it are called Rh negative. It has a great significance in pregnancy. Okay. If the mother have Rh negative and the baby in the womb be Rh positive, which is the possibility if the father be Rh positive. Okay. And then the Rh antibodies will be produced in the mother's serum, and such blood flowing through the placenta into the fetus may causes hemolysis and jaundice in the baby, endangering its life in the womb or soon after birth. The risk becomes greater in the successive pregnancy. Okay. Then study the blood clotting. Then the blood clotting. Blood clotting, when the blood is set, it soon loses its fluidity and red colored, soft jelly like sticky mass is formed. This mass blocks the broken surface of the blood vessels and avoid further loss of blood from the body. The soft mass thus formed is called blood clot, and the processes of the forming blood clot is called clotting of blood or coagulations of blood. The mechanism of the blood clotting is complicated one and involves many factors. The mechanism can be briefly described as below. You can see the mechanism of blood clotting. Prothrombin plus calcium plus thromboblastins is produce thrombin. Prothrombin is inactive, and then after that the process is the thrombin is active and from damaged cells or tissues. And after that then the thrombin plus fibrinogen is produced fibrin in fine threads. It's also inactive. Then the fibrin plus blood clots is produced blood clot. Okay, this is the process. Prothrombin plus calcium plus thromboblastin is produced thrombin, and then the thrombin plus fibrinogen is produced the fibrin, and after that the fibrin plus blood cells is produced the blood clot. Okay. It's all over the mechanisms of the blood clotting. Then whenever there is an injury to a blood vessels, there is formations of rough surface. The inner smooth lining of blood vessels is disturbed. After injury, you will know the blood vessels the blood vessels of the inner lining of the blood vessels are disrupted. When the blood platelets come in the contact with such a rough surface, they are injured. Due to injury, they release the substances called thromboplastins. Okay, then the injured tissues also secrete the thromboplastin. The injured tissues also secrete the thromboplastin. In the presence of thromboplastin and calcium, in the presence of thromboplastin and calcium in blood plasma, prothrombin, naturally occurring the plasma protein, is converted into thrombin. Okay, prothrombin, prothrombin plus calcium and prothrombin and calcium and thromboplastins is form the thrombins. Okay, which helps in the conservation of fibrinogen to fibrin. Okay, then the fibrin is insoluble and forms threads. Fibrin is insoluble and then it forms threads. The threads of fibrins are form a net. In the holes of this net, in cells are entangled. This mass then contracts to form a blood clot. The fluid that remain after clotting of blood is called blood serum. The normal clotting time of blood is 3 to 6 minutes. It is defined as the length of time between the moment of escapes of blood outside the vessels and the moment of development of fibrin thread. Okay, it is defined the length of the time between the moment of escapes of blood outside the vessels and the moment of the development of fibrin thread. Okay, and the time required for cessations of bleeding is called bleeding time. The normal bleeding time is 1 to 4 minutes. Factors hastening coagulations of blood are as follows: temperature slightly excess than the body temperature, contact of blood with rough surface and the injury of the blood platelets, excessive slowness of blood flow and the snake venom. Factors retarding coagulations of blood is low temperature, contact of blood with smooth surface and the presence of sodium or potassium citrate. Normally a blood clot is formed on the surface where the injury has occurred. When such a clot is formed within the circulations, it is called the thrombus. Whenever a portion of the clot gets detached and enters the circulating blood is called embolus. Okay, it is the factors retarding coagulations of blood and that is the factors hastening coagulations of blood. Okay, now disorders of blood. Major disorders of blood are anemias, polycythemias, bleeding disease, leukemias and agranulocytosis, thalassemia. Okay, now discuss one by one. First, anemia. Anemia signifies reduction in the amount of oxygen carrying hemoglobin in a given volume of blood. There are several types of anemia. In pernicious anemia, the intrinsic factors responsible for the absorption of vitamin B12 from gastric mucosa is absent. As a result, the number of erythrocytes goes down and their average size is increased. Size and the average size is increased. Hence, it is a type of macrocytic anemia. It is a type of macrocytic anemia. It contrasts to this in the case of microcytic anemia, the average diameter of the red is cell is smaller and the cause is mainly deficiency of iron. The clinical symptoms are similar to those of pernicious anemia. Hemolytic anemias are characterized by a certain lifespan of red cells. The resulting hemolysis, it may be either hereditary or because of circulating antibodies which becomes afferent of red cells. The heredity defect may involve abnormal red cells or abnormal hemoglobin. In sickle cell anemia, the defect is in the hemoglobin molecules. The shape of the large number of red cells is crescentic or sickle of their lifespan and considerable shortened. Okay, it is all about the anemias, and then we will discuss the thalassemias. Okay, in thalassemia, the hemoglobin defect results in a decreased lifespan of the circulating red cells. The shape of the red cells is altered, but a large number of nucleated cells are also observed. Many hemolytic anemias are caused by certain drugs, bacterial toxins, malaria, parasites, etc. Rh incompatibility can cause the hemolytic disease of the neuron. Okay, Rh incompatibility disease. Okay, hemolytic disease is called. The aplasia or atrophy of the bone marrow can cause the aplastic anemia, which result in the reduction of the number of the red cells, leukocytes and the blood platelets. Then polycythemia. Polycythemia refers to increase in the number of blood cells, especially the red blood cells. In the primary polycythemia, along with increased number of red cells, the bone marrow is also markedly hyperplastic. Skin and mucous membrane of mouth are red and the conjunctiva is red. In secondary polycythemia, there is a compensating increase in red cells in the conditions of sufficient oxygenations. Then the bleeding disease. The bleeding diseases are those in which the normal mechanism of the control of the hemorrhage is deficient or deranged. The bleeding time and the coagulations time are the tests used to confirm this disease. The hemophilia is characterized by a prolonged bleeding flowing a cord. It is heredity in nature. Okay, then the important features of the leukemia is proliferations of leukoblastic tissues resulting in a great increase in the white blood cells. It is the essential a cancer of bone marrow. Agranulocytosis. Agranulocytosis is a remarkable disappearance of granulocytic leukocytes from blood. It may be associated with the lesions of the mouth involving gums, tonsils, and even the bones of a jaw. Okay, that is the agranulocytosis. You will study the disorders of blood and and the blood groupings and also the blood clotting. Okay.